Hi guys, welcome back to Biology Videos at TDA. We're going to finish off Synapses, which is 13.5, so the second half, and then we're going to go to 13.6, which is Organisation of the Nervous System. So in this lesson we're going to recap Synapses. We're going to look at how we control synapses and the use of drugs and inhibitors, and we're going to look at what the body does and then artificial ways to change that, and then we're going to look at the... oh, a typo there, sorry. We're going to look at the organisation of the nervous system itself. So a quick reminder of an action potential. So when an action potential arrives at a presynaptic knob, we have the influx of calcium ions that go through the calcium channels into the presynaptic neuron. That then causes vesicles with neurotransmitters to migrate to the cell membrane. They then fuse, and that then causes neurotransmitters to diffuse across the cleft, binds to the sodium receptors if it's acetylcholine, binds to the sodium receptors, that causes them to open, that causes the depolarization. We then have the enzyme acetylcholinesterase that will then recycle the acetylcholine back, and that will repeat. Okay, so the control. So why are synapses useful? What can we have here? So if you look at these images here, you've got an image where you've got a neuron A and neuron B. You've got two neurons going into one. Okay. Now, and if you look at this first image, you've got some neurotransmitters, these green dots coming from A. You've got some neurotransmitters from B, and then you've got neurotransmitters from both. So what this allows in synapses is where we may have had just one action potential going down one neuron, we can then allow multiple to bind to one postsynaptic neuron, and that will allow control at that point. So we can, if we don't have a big enough stimulus from one, or from A, or from B, it might not cause an action potential in the postsynaptic neuron. It might require both. So these could be two separate stimuluses. So I'll make up something really crude. So it might be, you might see something scary, that might be A. You might hear something scary, and then this might be running away. So just seeing it alone might not be enough, hearing it alone might not be enough, but seeing and hearing something scary then causes a response. Okay? It also ensures that this unidirectional, so one directional travel of neurons is really important. And it also allows the opposite of this. It allows that one neuron can go in and it could go to multiple postsynaptic neurons. And we'll look at some images later on. So these are some of the benefits of having synaptic control. So how do we control the rate of action potentials between them? So we have something called temporal. When I think of temporal, I think of tempo, so the rate of something. So temporal summation, as a reminder, each action potential itself is exactly the same, and if it's in the same neuron, it will be at the same speed. It's whether it's myelinated in the diameter and the temperature that affects the rate of it. That's not what we're looking at here. Temporal summation is the frequency of the action potential. And you might need a certain amount of stimulus to create enough neurotransmitter to open enough channels in the postsynaptic neuron to cause this action potential. In this case, we don't have enough, so we've got low frequency, so we've got no action potential. Whereas in this example, we've got high frequencies, we've got multiple action potentials that are coming along. That causes so much neurotransmitter to then diffuse across the cleft, that then binds to the postsynaptic neuron, that causes an action potential. So it's many impulses from one to the other, and this will then increase the neurotransmitter concentration, and as we know there has to be a threshold, normally about minus 55 millivolts. This may then cause at this frequency the depolarization. So it might mean that a small amount isn't noticed, doesn't cause an action potential, but a large amount will do. So that's temporal summation. You've also got spatial summation, which we looked at kind of earlier on. Is the idea of you have multiple neurons going from one to the other. Again, we need this build-up of neurotransmitter. So it allows, spatial summation is multiple neurons into a synaptic, postsynaptic neuron, whereas temporal summation is the rate of them. And this image that I found from a textbook that's quite good kind of summarises that. And you can have both going on at the same time. You might have this really important neuron that's going to do a certain job, and you can have multiple sources of information that's coming along. So you've got excitatory, so you've got this E here, and then in the purple the I, inhibitory postsynaptic neuron, where the potential is produced. And you could have these purple ones are inhibiting the red ones, So you need a certain amount, and all of these mechanisms of control are going to be taken into account. There might be a really high rate over here and a low rate of inhibition, a high rate of excitation, which means that this will overcome. However, this might be a really high rate of inhibition over here, and these might be low. And then this will get to an overall action potential generation. So it might or may not reach the threshold of minus 55 millivolts. So it's really quite complex. You'll have all of these things going on to control the rate of the next. And you can kind of see how all these different mechanisms of control can cause us to do different responses. Now, once we get to these synapses, we have all these different neurotransmitters. So there's some of them you'll have heard before, so I've just thought I would summarise and show you some names. You don't need to learn all of these off by heart, but there's some that are likely to come up. Now, acetylcholine you need to learn. You also need to learn GABA, which is inhibitory. But there's some others that you might, these are amino acids, but there are some other you've probably come across before. Dopamine, serotonin, for instance, are quite common ones that you'll have heard of, which are often involved in the happiness response, how we feel. Now, as these neurotransmitters are chemicals, that means then that drugs can then affect that. So some drugs, such as strychnine, and this is an artist depiction, thinking about the 1800s of the results of strychnine poisoning. And a fun fact for you, I believe King Joffrey, I think his name was, I'm not a Game of Thrones fan, but apparently this is the poison that he received. So some drugs can cause various different things, such as overstimulation. And the way that strychnine works is it's a lethal poison that binds to and blocks glycine receptors. So the normal delicate balance between excitation and inhibition in the central nervous system is disturbed, and motor neurons will generate nerve impulses without restraint. So all skeletal muscles, including the diaphragm, they will contract fully and they will remain contracted. And because the diaphragm cannot relax, the victim cannot inhale and exhale, and therefore they're going to suffocate. And I think this image is trying to show that they are contracting all the muscles in their body. And this occurs in these Renshaw cells that you can see here within the nerve, and that causes this excitation of the motor neurons. Now you can have also, that's overstimulation, you can also have this excitotoxicity, so this word explaining that this can cause destruction. So this isn't this example, but this is another way that drugs can damage it. So an example where this can occur is with a high level of glutamate in interstitial fluid of the central nervous system. This causes the destruction of neurons through prolonged activation of the excitatory synaptic transmission. Now this can be found in things like stroke, and this lack of oxygen causes the glutamate transporters to fail. Then it will accumulate in the interstitial spaces between the neurons, and this essentially stimulates neurons over and over and over again. You don't need to learn that process, but the idea is it's this stimulation over and over and over and over again, and that causes damage to the neurons themselves, and it causes them to die, and that causes them in the brain, and then depending where it is in the brain, that will cause damage to the brain. It could be fatal, could be slightly damaging, could be repairable, but it could be really, really bad. In the textbook, you'll see some other examples. You don't necessarily need to learn them all off by heart, but what you do need to know are, what you need to be able to do is apply something to a real life scenario. So you might be told that you've got overstimulation of the skeletal muscles. What might that do? Well, the skeletal muscles are the ones that help us move around. It will stop us from moving. It also is the ones that help us breathe. That's going to be the key there. It's going to stop us from breathing. Okay, so that finishes off the synapses. We're now going to go on, it's quite brief, this section on the organisation of the nervous system. So we're going to have a look at that now. So this is the best image that I could find. I'll let you have a look at the joke as well. The best image I could find for this to summarise the nervous system, and there's two main sections to the nervous system. You've got the CNS, which you'll have heard of before, so the brain and spinal cord, and then you've got the peripheral nervous system. And if you learn what the word periphery means, it means on the outside. So the peripheral nervous system is essentially everything else, the control apart from the brain and spinal cord. So the peripheral nervous system, so this is new to A level now, will be split up into two sections. You've got the autonomic nervous system, and that's the word autonomic. Think of it as automatic. It's not the same thing, but it's the same sort of concept. The idea is that things are done without you having to consciously control them. and then you've got the somatic. So we'll stick with autonomic to start with. So the autonomic nervous system, the basic idea is, in our homeostasis of our body, we can increase or decrease the rate of something happening. So within the autonomic, we have the sympathetic and parasympathetic, and the sympathetic nervous system is a bit like our fight or flight. Our sympathetic nervous system gets us to increase the rate of things, so such as it might be increasing the rate of heart rate. And there's many nerves that go around the body, and one example is the sympathetic nerve and the parasympathetic nerve, often called the vagus nerve, which goes from the heart to the brain. And the sympathetic nervous system will speed up the heart rate, and the parasympathetic will slow down the heart rate. Okay? You've also got the somatic nervous system, and think of this as voluntary, conscious control. So this is looking at controlling of our skeletal muscles within our body. So we've got autonomic, unconscious, and we've got somatic, which is conscious control. And then finally, some examples that I've taken from the textbook to show you. So for instance, if we want to do anything sympathetic that's increasing the rate of things, essentially that's our fight or flight. What can we do to survive instantly now? So with saliva, we're going to need less saliva if we're trying to survive. We're going to increase the diameter of our bronchus. We're going to decrease urine. We're not interested in that. We're going to decrease peristalsis, is that movement of food, that churning, that squeezing of food around our intestines. So this is all going to be reduced, whereas the parasympathetic nervous system is going to increase those. So an easy way to think of the sympathetic and parasympathetic is it's our survival or it's our relaxation. And then finally, I just took the cut out from the textbook here. Sorry if the quality's not really good, it'll be better in the textbook. To kind of summarise it. So we've got our stimulatory idea from the skeletal muscle, from the somatic nervous system, making our peripheral muscles, skeletal muscles contract. And then you've got our autonomic nervous system, which is our smooth muscles. So they're the muscles that you don't consciously control. And that splits into sympathetic and parasympathetic. Okay. I've got a couple of questions with you here. The vast majority of the questions I could find on this were quite convoluted. There's lots of bits for you to read, so I'm going to email you them instead rather than put them all on here. But we do the first couple with me. So, have a go. And that was obviously A, so spatial summation, the idea of multiple stimuli. So a synapse is multiple going in and one response going out, and that's going to be spatial summation. Question 2, synapses are an integral part of the nervous system. Outline the roles of synapses. You don't need to explain in too much detail about how a synapse works, but the role, what is the function, and what are the things that kind of dictate and control it. Pause the video and have a go. Okay, so it allows this idea of cell signalling, it allows its communication, that's a role. It ensures it's unidirectional, so it's one direction that it can only go. It allows this convergence, divergence, so this idea of this spatial summation. It allows more than one neuron to be passed through a single one, and it allows one to be passed through multiple neurons, as we explained before around temporal and spatial summation. This idea that you need enough stimulation to cause the depolarization, so it allows this level of control, and it's kind of put in apostrophes there of background, is it ensures that a large enough stimulus is needed to cause a depolarization. And that can also prevent overstimulation as well, because of the mechanisms involved. It can also allow low level stimuli to be amplified as well, so you can go to multiple neurons. And this kind of idea that is inhibitory and stimulatory synapses allows control at the synapses. And then finally, and I apologise I haven't mentioned this before, but this is one of the ways that we learn and we build these circuits in our brain. If you think of a computer that can cause, if you do something you get a response, and that's one of the ways that our brains form memories in space and time, is they create circuits that when you're hit you then generate a memory. If anyone plays any musical instruments and you're trying to remember an old song that you've played, to start with you can't remember it, but as soon as you start kind of playing it, the circuit kicks in and then you can remember playing how to play the rest of the song. Okay, so the rest of it I'll email to you so you can submit as normal. Thank you very much. Good luck.